हेलो स्टूडेंट्स, कैसे हैं आप। मैं उम्मीद करता हूं कि आपने हमारी पुरानी वाली वीडियो वो देख ली है जिसमें हमने अल्फा हेलिक्स के बारे में डिस्कशन किया था कि अल्फा हेलिक्स है कैसे होते हैं। प्रोटीन की जो प्राइमरी स्ट्रक्चर है उसमें मैंने आपको एक्सप्लेन किया था द सीक्वेंस एंड द नंबर ऑफ अमीनो एसिड इन ए पॉलीपेप्टाइड चेन इज कॉल्ड एज द प्राइमरी स्ट्रक्चर द प्रोटीन। And we all know that the amino acids are bonded with the peptide bonds, as I have described in my previous video. The number of the amino acids are bonded in the linear fashion by the peptide bond for making a long polynucleotide chain. And such kind of the polynucleotide chain, जैसा कि मैंने आपको बताया, ये कभी भी स्ट्रेट नहीं हो सकती है। व्हाट इज द रीजन दैट ए पॉलीपेप्टाइड चेन इन द प्रोटीन स्ट्रक्चर cannot be in the straight line manner? Because of the tetrahedral nature of the carbon molecules and the angle of the rotation, as I have described into my previous video, the structure of the alpha helix. So I am requesting to you all that first of all, before watching this video, you have to understand the alpha helix structure in my previous video. This video is about the introduction to the beta pleated structure of the protein. As we know that the proteins are the building block of the life. More of the activities are controlled by the protein, as we all know that the enzymes are the catalyst which participating in the biochemical reaction to activate or to give the speed to the biochemical reaction by decreasing the activation energy. That the function of the enzyme makes the protein very important for our life and for the conduction of the biochemical reaction. This crucial role in this structure and the function of the cells. So at first we have to explain the beta pleated structure of the protein and what is the difference between the alpha helix and the beta pleated sheet. As I have explained you in my previous video, that whenever the sum of the amino acids are bonded with the peptide bonds in the linear manner, as I am making such diagrams, so many the amino acids which is you can see as the beads like structure. तो ये जो अमीनो एसिड्स हैं, all the amino acid has a chiral carbon, means the carbon alpha, as I have making this structure. The first group is the alkyl group and the carbonyl oxygen, and this structure is the peptide bond, and again the next structure is a second amino acid in a polypeptide chain. So many amino acids are bonded with the peptide bond, as you are watching in this structure, and this is the peptide bond. This is the peptide bond in the polypeptide chain, and I have already explained in my previous video that here between the amide and the alpha carbon, this bond they can rotate, and this bond also can rotate due to the stirring hindrance and due to the interaction of the alkyl group, as I have already explained in my previous video. So by the presence of the alpha carbon and the tetrahedral structure of the carbon and the angle of the rotation between the both side of the peptide bond, that's make the polypeptide chain in to acquire a particular 3D structure. Just like I have explained in the alpha helix. In the alpha helix, an polypeptide chain is coiled around an imaginary axis, just like a spring, the metallic spring. This is the alpha structure, and the alpha helix is stabilized, and this alpha helix is stabilized by the multiple hydrogen bonds between the carbonyl oxygen and the amide nitrogen, which provide the stability to the alpha helix. Here I am drawing a alpha helix because so you can clearly understand the difference between the alpha helix and the beta pleated sheets. This is the structure. This structure showing the alpha helical structure. And now what is the difference between the alpha helix and the beta pleated sheet? The first of all, I want to explain you that the polypeptide chains arranged on a plane in the zigzag manner. Just I have making this figure. The NS2, this is the N terminus of the polypeptide chain, and other is the carboxyl group. This is called as the C terminus. So now many polypeptide chains are arranged parallelly in this structure, parallelly arranged on a single plane, and these polypeptide chains are bonded with the hydrogen bonds, as the hydrogen bonds gives the stability to the alpha helix. Similarly, the carbonyl oxygen and the amide nitrogen makes the hydrogen bonds between these polypeptide chains and make a sheet-like structure. Such kind of the structure is called as the beta pleated sheet. So whenever it is asked what is the exactly difference between the alpha helix and the beta pleated sheet, so we can say in a alpha helix, a polypeptide chain coiled around an imaginary axis and gives the appearance just like a metallic spring to the alpha helix, and this alpha helix is stabilized by the hydrogen bonding, multiple hydrogen bonds between the carbonyl oxygen and the amide nitrogen of the amide. But on the other hand, in the beta pleated sheet, as you can see in this figure, in the beta structure, the polypeptide chains arranged parallelly to each other and making the hydrogen bonds in a single plane. This structure seems like the plate-like structure, a sheet-like structure. That's why it is called as the beta sheet. And why do we say it the pleat structure? Because all the polypeptide chains are making a zigzag structure. That's why we are saying this structure as the beta pleated sheet. So this is the reason. Whenever it is asked that what is the reason of the secondary structure, then we can say that by the presence of these angle of the rotation between the peptide bond and the amino acids and the tetrahedral nature of the carbon atoms, these are the two main factors that a polypeptide chain cannot remain in a state-like manner. So, for acquiring a proper conformation, these polypeptide chain, newly synthesized polypeptide chain, and acquire a specific 3D structure or make the secondary structure. So, I have already explained in my previous video that what is the mean of the secondary structure. Secondary structure means the conformation of the backbone. Conformation of the backbone of the polypeptide chain is called as the secondary structure. So let me explain in the next slide. The secondary structure, the alpha helix and the beta pleated sheet. The protein can adopt two secondary structures. The first is the alpha helix and the beta pleated sheet. I have already explained in the previous video. And in the alpha helix, the polypeptide chain coiled in around an imaginary axis. I have already explained you. And the polypeptide chains arranged side by side, the parallelly on a plane, a common plane. This is the beta pleated sheet. The Linus Pauling and the Robert Corey, a famous biochemist, discovered this structure, the alpha helix and the beta pleated sheet, by the X-ray crystallography technique in the 1951. Whenever it is asked in the objective examination that which technique is used to elucidate the structure, the 3D structure of any biomolecule, then you can say the X-ray crystallography technique. The X-ray crystallography technique is used to we can say visualize the 3D structure of any biomolecule. So it may be asked into the objective examination and the multiple choice questions. This name the pleated, why we are using the pleated because the polypeptide chain backbone adopts a zigzag arrangement. I have already explained you. So here in the figure you can clearly see that there are two kinds of the secondary structure a polypeptide chain can acquire, that is the alpha helix and the beta pleated sheet. So understand the exact basic structure or the basic feature of a polypeptide chain in the beta pleated sheet. Here you can clearly see in this figure this image. This image is showing the beta pleated structure, and what is explaining I am here that as you can see this beta pleated sheet, as you can see in this figure, this is the backbone of a polypeptide chain. Here this backbone makes the zigzag arrangement. Here as I am drawing in this image, this backbone of the polypeptide chain acquire a zigzag arrangement, and similarly Similarly, in the other polypeptide chain, you can also see the zigzag arrangement. I am making the black linings to discriminate this structure from the rest of the chemical structure. These are the backbone of the polypeptide chain, and the polypeptide chain makes the zigzag arrangement. Okay, and two polypeptide chains are arranged, and here what you are looking in this figure, this is the carbonyl oxygen. This is the carbonyl oxygen. What is this carbonyl oxygen? I can draw here a separate figure, just like this is a alpha carbon of an amino acids. I am drawing either the alpha carbon, and other molecule is the carbonyl oxygen, C double bond O, and this is the amide group of the peptide bond, NH, and again another carbon, alpha carbon, and again here I am drawing the C double bond O. This is the carbonyl oxygen and the amide. This is the polypeptide chain when polypeptide chain acquire a zigzag structure. And similarly, if I draw another polypeptide chain, so here I can make another polypeptide chain. This is the carbonyl oxygen, this is the nitrogen and the hydrogen, and this is a carbonyl oxygen, and this is the NH2. Here I am drawing another polypeptide chain in this structure. You can see that whenever the carbonyl oxygen of a polypeptide chain comes near to the hydrogen of a amide of a polypeptide chain, this makes the hydrogen bonding between these two polypeptide chain. This is the hydrogen bonding. Whenever hydrogen lies between the two high electronegative molecules, this hydrogen makes a weak bond, hydrogen bond. So in such like manner, whenever two polypeptide chains arranged parallelly in the zigzag manner, the carbonyl oxygen of one polypeptide chain and the amide hydrogen of the amide of another polypeptide chain makes the hydrogen bondings between these two polypeptide chains. And each polypeptide chain comprising 5 to 10 amino acid. In this structure, in each polypeptide chain, how many amino acids are present? Then we can say that 5 to 10 amino acids are present in a polypeptide chain on an average. And how many polypeptide chains may be present in a beta sheet? Then we can say that 2 to 15 polypeptide chains, 2 to 15 polypeptide chains can make a common beta sheet, sheet. Here you can also read that polypeptide chain are the extended and the 2 to 15 adjacent beta strands align in a sheet-like arrangement, and each polypeptide chain comprising the 5 to 10 amino acid residues. And each strands adopted the zigzag extended conformation. I have already explained. That's why we say this sheet as a beta pleated sheet. I have already explained about the hydrogen bondings. You can read here in these 2 points. These hydrogen bondings stabilize the beta structure. ये क्या करती है कि बीटा स्ट्रक्चर को मजबूती देती है और उसको एक हम कहेंगे नेचुरल कन्फर्मेशन देने का काम करती है. आगे देखेंगे आप इस स्ट्रक्चर में ये क्या है. The types of the beta sheet. Actually, what is the mean of the types of the beta sheets? This means the arrangement of the polypeptide chain in a different beta sheets. Here I am making another diagram. In the antiparallel beta sheet, what is the mean of the antiparallel beta sheet? When a polypeptide chain, when two polypeptide chains are arranged in such a manner that the N terminus of each polypeptide chain comes near to each other and the C terminus near to each other. Such kind of arrangement is called as the parallel arrangement. Such kind of the arrangement is called as the parallel arrangement कहते हैं. Parallel beta sheet बोलते हैं. बीटा शीट का मतलब क्या हो पैरेलल? जब कभी भी दो पॉलीन्यूक्लियोटाइड चेन होते हैं उसके एन टर्मिनस एक दूसरे के पास में हो और दूसरी तरफ आप देखेंगे सी टर्मिनस है वो एक दूसरे के पास में हो, तो ऐसी जो चेन होती है उसको हम बोलते हैं पैरेलल बीटा शीट. On the other hand, when the N terminus of a polypeptide chain when N terminus of one polypeptide chain remains near to the C terminus of another chain, यानी कि एक दूसरे के अपोजिट डायरेक्शन में है। आप देख पाएंगे इस चैन का N टर्मिनस दूसरे के C के पास है, इसका C टर्मिनस है वो N के पास है। ऐसा जो अरेंजमेंट होता है उसे हम क्या बोलेंगे? इसे हम कहते हैं एंटी पैरेलल बीटा शीट बोलते हैं, एंटी पैरेलल बीटा शीट कहते हैं। बिल्कुल सिंपल सा है, पैरेलल और एंटी पैरेलल सेम एंड्स एक दूसरे के पास में तो पैरेलल हो गया, और अपोजिट एंड एक दूसरे के पास में है तो एंटी पैरेलल कहते हैं। और कई बार ऐसा भी होता है कि कुछ ऐसी स्ट्रक्चर्स होती है उसमें मिक्स्ड अरेंजमेंट होता है। मिक्स्ड अरेंजमेंट का मतलब क्या हुआ कि एक ही स्ट्रक्चर में, जैसा कि मैं यहाँ पर आपके सामने बना रहा हूँ, आप इस फिगर को देख पाएंगे। अगर यहाँ पर मैं 3 पॉलीपेप्टाइड चेन बनाता हूँ तो यहाँ पर आप देखेंगे कि क्या मैंने डिफरेंशिएशन किया है। तो यहाँ पर आप ये देख पाएंगे क्लियरली कि यहाँ पर फर्स्ट और सेकंड जो पॉलीपेप्टाइड चेन है इसके सी टर्मिनस एक दूसरे के पास में और एन टर्मिनस है वो एक दूसरे के पास में। इसका मतलब ये कैसा अरेंजमेंट है? ये है पैरेलल। लेकिन सेकंड और थर्ड पॉलीपेप्टाइड चेन की बात करेंगे तो यहाँ पर आप क्या देख रहे हैं? सी के पास में यहाँ पर है एन टर्मिनस, इसका मतलब ये क्या है? एंटी पैरेलल। अगर किसी सिंगल बीटा प्लीटेड शीट में दोनों तरह का अरेंजमेंट होता है ना, तभी हम उसको क्या बोलते हैं? मिक्स्ड बीटा शीट बोलते हैं, मिक्स्ड बीटा शीट कहते हैं। बट द क्वेश्चन इज विच अरेंजमेंट इज द मोर स्टेबल? Which arrangement is the more stable? Then we can say that anti-parallel arrangement is the more stable conformation of the beta pleated sheet. Then what is the reason behind it? That you can see in the second point. In the anti-parallel beta sheet, these hydrogen bonds are nearly linear 180 degree, making them stronger. एंटी पैरेलल में क्या होगा? जितने भी हाइड्रोजन बॉन्ड्स होंगे वो एकदम स्ट्रेट रहेंगे, उससे क्या होगा कि इसमें स्टेबिलिटी आती है। जबकि पैरेलल अरेंजमेंट में आप देखेंगे मैंने क्या लिखा? हाइड्रोजन बॉन्ड्स आर द एंगल्ड। किसी एक का हाइड्रोजन बॉन्ड ऐसे है तो किसी दूसरे का अलग एंगल है, अलग डिफरेंट एंगल में है, जिससे क्या होगा कि पैरेलल अरेंजमेंट होता है वो मोर स्टेबल नहीं होगा एज कंपेयर टू द एंटी पैरेलल बीटा शीट। In the mixed contain both the parallel and the anti-parallel arrangement, and such kind of the arrangement is found in the beta barrel and the beta sandwich motif. And what is the mean of this beta sandwich motif? You can clearly understand into the next slides. So here this figure is also showing the anti-parallel arrangement of polypeptide chain. Here you can see it is written that anti-parallel arrangement means the N terminus of one polypeptide chain is just near to the C terminus of the another, and similarly on the opposite end the C terminus of a polypeptide chain is present near the N terminus of the other chain. Such kind of the arrangement is called as the anti-parallel arrangement. And another important thing is that these hydrogen bondings, the hydrogen bonding between the carbonyl oxygen, here I can draw that hydrogen bonding of carbonyl oxygen of a peptide bond and the hydrogen of the amide group of another polypeptide chain. Similarly, we can draw a simple structure. This is the polypeptide chain first, and this is the second polypeptide chain. When two carbonyl oxygen and the hydrogen of the amide of a different another polypeptide chain comes to near, this makes the hydrogen bondings. This is the hydrogen bondings between the polypeptide chains. So these are the hydrogen bondings. I can draw here so that you can clearly see. These are the hydrogen bonds. These are the hydrogen bondings between these two polynucleotide chains. These are the hydrogen bondings, and these all hydrogen bondings are at the 180 degree, just at the parallel. All the hydrogen bonds are nearby parallel to each other, which makes the anti-parallel arrangement a stable conformation. Then I am here going to explain So what about the orientation of the alkyl group? What is the arrangement of alkyl group arrangement, and which factors stabilize the structure of the beta sheet? So we can draw another diagram. Here I am making a polypeptide chain NS2, and this is the carbon alpha of first amino acid, and here this is the carbonyl oxygen, and this is the amide group of peptide bond, and another this is the carbon alpha of second amino acid, and here I am making the carbonyl oxygen and again the amide hydrogen. This is the arrangement of a polypeptide chain. But here I am talking about the orientation of the alkyl group. Just imagine this is the alkyl group. This is the alkyl group of a first amino acid, and this is projected out of the plane. This is projected out of the plane, and the alkyl group of the next amino acid is projected opposite to the we can say the other side of the plane of the paper. The alternative arrangement, this arrangement is found alternatively if the alkyl group of one amino acid is out of the plane of the paper, then the alkyl group of the next amino acid projected downward to the plane of the paper, and similarly the alkyl group of the third amino acid is projected again out of the plane of the paper. So we can say it is a alternative arrangement. I am making another diagram to explain you. Here if I draw this structure is the beta pleated sheet. This is a beta pleated sheet, or we can say this is the plane of the paper. This is the plane of the paper, and here this is the carbonyl oxygen, and at the plane of the paper this is the NS2 group, and this is the carbonyl oxygen. Then here what is the arrangement of alkyl group? This alkyl group projected outside the plane of the paper. Yeah, this is the alkyl group which is present at the outside the plane of the paper, and this is the carbonyl oxygen, and again I am making here the amide hydrogen, and again this is the carbon alpha of another amino acid, and the alkyl group of this group is downward projected from the plane of the paper. तो अगर आप ये मान लेते हैं कि we are considering this is the plane of the paper, then first alkyl group is projected outside the plane of paper and the next alkyl group is projected downward, and similarly next third alkyl group again projected upward. So here what is written in this slide, the side chain, the alkyl group of the amino acid project alternatively above and below the sheet, reducing the steric hindrance and giving a pleated appearance. अब आप consider कीजिए, if you are consider that all the alkyl group, if the all alkyl group is projected upward. Say this is the structure, and this is the second amino acid, and this is the alkyl group of the second amino acid. In that condition, it creates a steric hindrance between these two alkyl groups. This makes this structure unstable. This is the reason that alkyl group of one amino acid projected upward and the alkyl group of another amino acid is downward. And another reason, if we consider the alkyl group of two adjacent amino acids are positively charged, in that condition they can repel to each other, and if these amino acids are, the alkyl group is the negatively charged, this also makes the repulsion in the alkyl groups. So to decrease the repulsion and to decrease the steric hindrance between two alkyl group of the adjacent amino acid. These alkyl groups are arranged alternatively. So in this structure, what I am going to explain you about the stability of the beta pleated sheet. The first important factor is hydrogen bonding pattern. As I have already explained that in the alpha helix, the beta pleated sheet is coiled around an imaginary axis, and this alpha helix is held by the so many hydrogen bonds between each first and the fourth amino acid, similarly second and the fifth amino acid. Similarly, in the beta pleated sheet, all the polypeptide chains are bonded to each other by the numerous hydrogen bonds, which makes the beta sheet stronger. So hydrogen bonding the patterns makes the beta sheets a stronger structure. And other important factor is the distribution of hydrophobic and the hydrophilic residues. All the hydrophilic residues. And what is the mean of the hydrophilic residues? The amino acid which has the charged alkyl group, just like the aspartic acid and the glutamic acid. In my previous video, I have already explained about the hydrophilic and the hydrophobic amino acid. Just like the aspartic acid, the alkyl group of aspartic acid is CH2 and CO minus, and similarly the CH2, CH2 and COO minus. This is the alkyl group of the glutamic acid. And here I am making another alkyl group of the lysine. This is CH2, 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 4 times CH2, and after that an NH3 plus. So such kind of amino acids are the charged or the polar molecule which makes this amino acid as the hydrophilic residues which can attract toward the water. So such kind of other residues and the hydrophobic residues. And what is the hydrophobic residues? Just like the valine. Here I can make the alkyl group. In the valine, there is CH and the CH3 in place of the alkyl group. And similarly in the leucine, the CH2, CH and CH3, CH3, this is the leucine. Such kind of the amino acid having the long aliphatic chains behave like the hydrophobic. So distribution of hydrophilic and the hydrophobic amino acids in a beta pleated sheet interfere the stability of the beta pleated sheet. And third important point is the presence of small amino acid residues, just like the glycine, alanine and the valine. The glycine, as you know, this is the smallest amino acid. I can draw here. Here in the glycine, only hydrogen is present in place of the alkyl group. This is the structure of the glycine. In the glycine, only hydrogen present in the place of the alkyl group. It makes this is the lighter lighter amino acid which has the least molecular weight. And another amino acid just heavier to the glycine, this is the alanine, where a methyl group is present in place of the alkyl group CH3. So glycine and alanines, like the small sized amino acids, also make the stable to the beta pleated sheet. So here I have made some of the points which amino acids are the preferred for the beta structure and which are unfavorable for the beta pleated structure. Just like the favorable residues, amino acid residues like the glycine, alanine, valine and isoleucine, these are the favorable amino acid for making a beta pleated sheet. On the other hand, the unfavorable amino acids are the proline. Proline induce the kink. And what is the mean of the kink? Kink means the fold. Kink means we can say a dent in the polypeptide chain which disrupt the hydrogen bonding. Why? Why the proline make the kink? Because we know the proline has a pentagonal ring structure in which the amide nitrogen is the part of this pentagonal structure, which makes a kink. or we can say the proline is a band producing amino acid because of the nitrogen of amide is the part of the pentagonal ring. I have already explained in detail in the previous video the types of the amino acids and in the alpha structure of the protein. Similarly, alternation of the polar and the non-polar residues often occurs leading to the amphipathic sheet. We can say in the simple language that some of the beta sheets contains both kind of the amino acids, the hydrophilic and the hydrophobic. If a molecule has both the nature, hydrophilic nature as well as the hydrophobic nature, such kind of molecules is called as the amphipathic. So, beta pleated sheet may be the amphipathic in nature if consisted of hydrophilic and the hydrophobic amino acids. And these are the biological importance of the beta pleated sheet. I am just read out these points. The structural role. What is the structural role? You can understand the provide rigidity to the protein and the tensile strength. इसको strong मजबूत देता है. Just like the silk fibroin. Whenever it is asked into the examination which kind of the secondary structure is found in the silk fibroin, then you can say the beta pleated sheet. Every the sentence make a objective question, so you can clearly understand and learn. Whenever it is asked in the multiple choice examination, which kind of the secondary structure is found in the silk fibroin, then we can say the beta sheets. Beta sheets are core element of many globular protein. And other function is the functional role. What is the functional role of the beta sheet? Found in the enzymes. So many enzymes consisted of the beta sheets, the transporter proteins, which functions like the carrier molecule, also comprises the beta pleated sheet and the signaling protein. Beta sheet is also a special component of the signaling protein. The beta barrel from the membrane channel and the transportation ports. These are the some of the pathological role of the beta pleated sheet. And what is the pathological role? Here I am saying that whenever a protein misfold due to the misfolding in polypeptide chain or misfolding in the protein structure, this makes a protein inactive, and that disrupted the protein create the different disease like the Parkinson and Alzheimer disease. These diseases are created by the a disrupted structure of the protein, or we can say it's created by the misfolding of the polypeptide chain in a protein. Just like this is an important example. The misfold, the beta sheet aggregates into the amyloid fiber. All in this disease, in the Alzheimer disease, if whenever it asked what is the reason, biochemical reason behind the Alzheimer disease and the Parkinson disease, then we can say that the beta sheet, misfolded beta sheet aggregate into the amyloid fiber, which create the Alzheimer disease or the Parkinson disease. And another, the beta sheet propensity of certain sequences underline protein aggregation disorder. This disrupted the protein or the misfolded protein the aggregate into a particular tissue or the part of the body and create different kinds of the disease. Here, these are the special modification of the secondary structure. So these structure is also called as the super secondary structures. These are called as the super secondary structure. Very important point. A super secondary structure. And what is the mean of the super secondary structure? As its name implies, these are the secondary structure. But why do we say that this is the super? When two or more kind of the secondary structures assembled to each other and makes a large complex, such kind of structure is called as the super secondary structure. What am I saying? That you can already understand that there are two types of the secondary structure. The first is the alpha helix and other is the beta pleated sheet. Okay, when these alpha helix and the beta pleated sheet assembled to each other and if arranged just like beta, alpha and beta. If I want to draw something about this structure, just this is the beta sheet and this is another beta sheet, and when these beta sheets are joined to one another in this manner, this is the alpha helix, this is the alpha helix. We can say the both the beta pleated sheets in this structure are joined by the alpha helix. So here we can say the three different secondary structures, the one is the beta, second is the alpha, and the third is also the beta structure, makes and assembled to each other and makes a large complex. This is called as the super secondary structure. So there are so many types of the super secondary structure as written here: the beta hairpin, beta meander, beta barrel, and the beta sandwich. Beta hairpin, I am explaining these all structures, these all super secondary structures into the next slide. With the example and the diagram, you can clearly understand. This is the super secondary structure. Which one? The hairpin structure. This is called the hairpin structure. And what is, you can see in this structure that in this figure, these are the two beta sheets. This is the one beta sheet and this is the another beta sheet. And you can clearly see that these chains are the antiparallelly arranged. This arrangement is called as the antiparallel arrangement. As I have already explained, when the N terminus of one polypeptide chain comes near to the C terminus of another polypeptide chain, such type of arrangement is called as the antiparallel arrangement. In this structure, the hairpin structure, two beta pleated sheets, two parallel antiparallel beta sheets are connected by a short loop of 2 to 5 amino acids. Here 2 to 5 amino acids are present in between the connection site of these polypeptide chains. And such kind of super secondary structures are commonly found in the globular protein. So what is the significance of these structure? These structure, just like the hairpin structure, helps in the folding and provide the stability to the protein. And another, this facilitate the protein-protein interaction and also gives the diverse biological functions, just like this such kind of the structure is found in the antimicrobial peptides and the beta barrel protein, and in many enzymes such kind of hairpin structure is found. Then which kind of the examples here, you have to understand that C2H2 zinc finger protein, this is a special kind of the protein, C2H2 zinc finger protein. In this protein, hairpin is the common secondary structure, or we can say the hairpin is in a common super secondary motif in the DNA binding domain of this protein. Similarly, in the antimicrobial peptide, it is a common motif of this enzyme which facilitate the disruption of microbial membrane. These are the specific function of the super secondary structure. The next is the beta meander. What is the beta meander? At first, I am saying that you have to focus on this first diagram. The first beta sheet and this is the second beta sheet. Both are arranged in the antiparallel manner, and this is the third, this is the fourth, and this is the five. The five beta sheets antiparallelly arranged and joined together by a particular 3 to 5 amino acids band. Two or more antiparallel beta strands linked by the hairpin loops, and it is frequently found in the large beta sheet. And one of the important example of this beta meander is the pCNA. What is the pCNA? The proliferating cell nuclear antigen. This is the antigen which is called as the proliferating cell nuclear. This antigen contains the beta meander super secondary structure. And you can clearly see in this secondary structure, which is showing that these two beta sheets, first and second, these are the beta sheets are arranged parallelly to each other, because the C terminus of these polypeptide chains are near to each other and the N terminus is also near to each other, which makes this arrangement the parallel. And the alpha helix, this is the alpha helix structure, this is the alpha helix structure. The one end of the alpha helix is joined from the one polypeptide chain and the opposite end of the alpha helix is joined with the another polypeptide chain. This is called as the beta alpha beta, because this is the beta alpha and the beta, three secondary structures are connected. It is the fundamental block of many proteins, just like the ribonucleases. ये जो ribonucleases होते हैं enzymes, ribonucleases enzyme comprises of the beta alpha structure, and another important enzyme which is the triose phosphate isomerase. Triose phosphate isomerase. This enzyme is used in the glycolysis. This enzyme, which is a protein, also contain such kind of the super secondary structure. The alpha helix is positioned above the plane of the parallel. कैसे देखते हैं कि कैसे अरेंज होता है इसमें. यहाँ पे मैं अगर आपको ये समझाना चाहूँ तो मैं ये कहूँगा कि ये वाली जो स्ट्रक्चर है ये है एक बीटा शीट है. सिमिलरली यहाँ पर एक और अनदर एक है बीटा शीट है. और जब मैं आपको ये बताऊंगा कि इसमें अल्फा हेलिक्स कैसे रहता है, अल्फा हेलिक्स का अरेंजमेंट रहता है ऐसे ऊपर की तरफ रहता है. यहाँ पे ये कनेक्टेड यहाँ पर है, ये कनेक्टेड यहाँ पर है. इस प्लेन से बीटा प्लीटेड शीट के प्लेन से क्या रहते हैं, ऊपर की तरफ रहते हैं. दिस इज एन अनदर सुपर सेकेंडरी स्ट्रक्चर दैट इज कॉल्ड एज द बीटा सैंडविच. Sandwich, what is the mean of the sandwich? When two the plane structure lined or arranged to each other, the two antiparallel beta sheets, you can see in this figure there are two antiparallel beta sheets twisted to each other. The two beta sheets face to face packed in a layered arrangement for making the sandwich. The layered arrangement with a gap of the 10 angstrom. So we can say the two beta sheets are arranged in the layer format, just like, and there is a difference between the distance between these polypeptide chain is the 10 angstrom, and these are the antiparallel arrangement of the beta sheet. This is making the beta sheets up antiparallel, and these structures are twisted to each other, just like in the figure first. This motif helps in the protein folding and the interactions. The example of these beta sandwich, whenever it is asked which one is the best suitable example for the explaining the beta sandwich structure, then we can say the beta immunoglobulin IgG. And this IgG consisted of 7 to 9 antiparallel beta sheets, 7 to 9 antiparallel beta sheets, and another is transthyretin. This protein helps in transportation of the thyroxine and the retinol. As its name implies, the transthyretin, जो वर्ड है वो किससे बना है, थायरे और रेटिन दो से बना हुआ है. थायरे का मतलब हो गया थायरस यानी कि थायरोक्सिन, और रेटिन हो गया रेटिनॉल, इससे बना थायरेटिन. ट्रांसथायरेटिन प्रोटीन है, वो हेल्प करती है ट्रांसपोर्टेशन ऑफ द थायरोक्सिन एंड द रेटिनॉल. एंड थर्ड इंपॉर्टेंट एग्जांपल इज द टेनासिन. ये क्या है, द थर्ड फाइब्रोनेक्टिन टाइप थर्ड रिपीट्स इन द ट्रांसिन हेल्प्स इन द सेल एडिशन में ये हेल्पफुल रहता है. तो ये डिफरेंट टाइप की क्या है, सुपर सेकेंडरी स्ट्रक्चर है. एंड दीज आर द इंपॉर्टेंस of the beta pleated sheet in the protein structure. However, I have explained already in the previous slides that beta structure provide the stability. I am not I don't want to read these points. You can note down these points, or you can take the screenshots and note down these importance of beta pleated sheet, the molecular binding and the mechanical support, just like in a structural protein like the silk fibroin and the collagen fiber, beta sheet provide the necessary tensile strength. We can understand the silk fibron is a very strong fiber. This strongness is provided by the beta sheet, and the collagen filament are very important and the very strong filament. These collagen is also made up of the beta sheet, which provide the tensile nature and the strong nature, or we can say provide the mechanical support to the body. These are the conclusion and the key takeaways. In the summary, the beta pleated sheet is a crucial secondary structure in the protein, conferring the exceptional stability, rigidity, the versatility, and its a unique hydrogen bonding and the hydrophobic interaction enable diverse biological function. So in conclusion and in the summary, I want to explain that uh, the first important thing is what is the difference between the alpha helix and the beta pleated sheet. In the alpha helix, only a single polypeptide chain coiled around an imaginary axis, and in the beta sheet, so many polypeptide chains are arranged on a plane of the paper. This is the basic difference. And what is the similarity? In the alpha helix and in the beta pleated sheet, in both the structure, the stability, the conformation of polypeptide chain is held by the hydrogen bondings between the carbonyl oxygen of one polypeptide chain and the amide hydrogen. Thank you. Thank you so much.